All right, welcome to the real part two. I think in the last video I said that was part two. This is actually part two. I'll sort that out in the intro. I've yet to make the intro to this lab, but yeah, this is part two, the quantitative analysis. I was calling the other one part two, saying I'll see you in part three, but anyways, that was part one. This is part two, quantitative analysis of biological molecules. In this case, you're going to set up a series of dilutions. And what we're going to use is red food dye. So you're going to easily see your dilutions and how you do with the dilutions. So in this part of the lab, this is when you use the larger test tube rack. That's when you use the larger glass test tubes. So I think in the handout it tells you to gather these and label them. And we're going to have them labeled for you. So you should see on there, it should say 1, 2, 3, and 4. So make sure they're in the correct order so that you know what goes where. So in this section also you're going to use these other pipettes. So instead of using the transfer pipettes, you're now going to use the graduated pipette. So the graduated pipette allows you to have an exact amount that you're pipetting. And you'll notice there are two measures on it. So on one side it's going to count up in volume, and on the other side it's going to count down in volume. So this allows you to do two different things. If you want to, you can draw up, say, 10 milliliters, because this is a 10 milliliter pipette. You could draw up to 10 milliliters, which is going to have you up here at the 0. And if you wanted to dispense 4 milliliters of liquid, you would go down to 4. So you've dispensed 4 milliliters of liquid. Alternatively, you can do what I like to do, and that's to use the ones that go up, so that are increasing volume as you go this way. To me, it's just more straightforward. I only like to draw up what I'm going to dispense. So if, say, I wanted to draw up 8 milliliters of water, I would draw up 1 milliliter, 2 milliliters, 3, 4, 5, 6, all the way up to 8. And then I have only 8 milliliters in my graduated pipette, and then I can dispense that into my test tube. So just pay attention that you do have two sides. Again, I recommend using the side that counts up from 0 to 10. So the way this one works is you have to make sure that you have your pipette pump and your pipette, and you have to make sure it's firmly seated into there. You don't have to shove it all the way up in there, but you don't want to be too gentle because it's going to fall out or it's going to get air up in there. You need to make sure that's firmly seated in there, and then you also want to make sure all the air is like expelled out. So same as with the transfer pipette, firmly seated, air expelled out. So if this is set like this, it's not ready to go. This pipette pump needs to be set back to 0, which is to do that. Because what's going to happen is, the way this works is with suction, you're going to roll this wheel. If you roll the wheel kind of down towards your liquid, it's going to pull this part up, which is going to create suction and draw the liquid up. So this here is the trigger to release your liquid though. So you want to make sure that you're not holding that while you're drawing it up, or you're not going to draw anything up because it's going to be not going to be able to pull the liquid. So some of them don't have this trigger. The way you operate them is just one way goes up and draws the liquid in, and the other way you roll the wheel the other way or push up here and it'll dispense it down. Same as with the transfer pipette, you don't want to operate things too quickly or it'll splash everywhere. So if you pull it up and then just try to slam that down, you're just going to splash water all over, or whatever your liquid is, all over the out of the test tube and such. But so we'll go over here and I'll show you how to use this in a bit more detail. Again, what you're going to do if you're following along with your handout is you're going to have your 4 test tubes. Into tube number 1, you're going to put 8 milliliters of water. And then into tubes 2, 3, and 4, you're also going to put water in those. But in this case, it's going to be 4 milliliters of water. So let's go over here. Again, the water is going to be on the side, just like it was in the qualitative analysis. You can use that same beaker of water that you were using from qualitative analysis, but instead of the transfer pipette, you're going to use the graduated pipette. So, as I said, I need to have this ready. Make sure the pipette's firmly seated into the pipette pump. Make sure it's set to 0, all the air's expelled out. Tip is going to go down below the water before I even do anything. So you got to get that down below the liquid. It doesn't have to be all the way down to the bottom, but you want to be somewhere close to the bottom. And then I'm just going to start rolling this up. Again, making sure that my finger's not on the trigger. So I'm rolling that up. and you'll see us drawing liquid up in. And like I said, I like to use the side where I can see the volume going up to the amount that I want. So I've drawn up 8 milliliters of water. So I have 8 milliliters of water there. Now I'm going to bring it over here to my first test tube. And again, I have really 3 different ways I can dispense it. I can press the trigger. If you tap it a little bit, it'll go slowly. Or you can roll it down with this. Or what some people do is they use this up here. I don't like using that as much, but it does work. I like to roll it. If you're using the trigger, it doesn't always get that last little bit out. You know, so there's a tiny little bit of water in there. Just finish rolling it down with this, and it'll get the last of it out. So that was 8 mils of water. You're going to do the same with 4 mils to here, 4 mils to there, 4 mils to there, and so forth. Then after you have your 8 milliliters of water in test tube 1, and then 4 and 4 and 4 in those, you're going to add 1 drop of red food coloring to your first tube. And only to your first tube. You don't add food coloring to the others. Again, firmly seated, depress the air out, and you only need 1 drop. It's like a little air bubble there, but I'll squeeze it till we get 1 drop. And you only need 1 drop. If you get more than 1 drop, it's not going to work. So 1 drop there. And then we're going to go back over here. You got to mix it up. So finger over the end firmly, mix it up. And then you're going to set up your serial dilution. So you're going to make 2-fold dilutions. So the way you're going to make 2-fold dilutions is you're going to take 4 mils from this and add it to this tube with 4 mils. And then so forth. So again, you're taking your volume, your concentration, and you're diluting it 2-fold. So if you had a concentration of 1 drop of food dye per 8 milliliters of water, and then you take that concentration of food dye and add more water to it by taking some part of this and putting it into that test tube, you've now diluted your concentration. So instead of having 1 drop of food coloring per 8 milliliters, you now have something else, and that's part of the lab is for you to figure that out. So we ask you that, think about that. What would it be if you do a 2-fold dilution of that? What does it end up being? And then you're going to do the same thing for the rest of these 2-fold dilutions by, again, taking out 4 mils, adding it to the next tube, and then mixing it well, taking out 4 mils, adding it to the next tube. Again, each time you're diluting it, diluting your food dye. And that's part 2 of the lab, setting up these serial dilutions. In part 3, we're going to test and see how well you did with those dilutions. Again, if you started off with the right amount of food dye, and then you diluted it properly without mixing, getting too many drops where they're not supposed to be, and using the right volumes with your graduated pipette, we should be able to see that, and we're going to use a instrument to read and see how well you did. So that'll be part 3. I'll be back over here telling you about the spectrophotometer.